thank you very much for having me, even over Zoom. I did not expect that to be that well organized, so thank you very much for that. And I'm sorry I couldn't join physically, but next time. So this talk is going to be less technical, I think, so maybe a bit a lighter topic, but hopefully still interesting. And I'll be talking about the pH-dependent cluster of charges in a conserved cryptic pocket on Flaviviral envelopes, which is a long title. So I'm going to go through the segments and explain the bits. And also, if you want spoilers, feel free to scan the QR code. Otherwise, I'll just proceed. So first of all, I'll be talking about the virus. So it's going to be a long introduction, I'm sorry, but I think it's important to set the stage so actually you know why do we even need to care. And specifically, why do we want to look into these pockets? So there's going to be some talk about the pockets, but then so it's going to be a long-winded intro without any electrostatics, but then there's going to be a payoff. So there's going to be a cluster of charges that comes into play. So just bear with me. All right. So when we talk about fly viruses, there are some viruses you might recognize from this list that belong to that viral family. Dengue virus, for example, is a most common example. Zika virus that kind of come to prominence during Brazilian Olympic Games. There's a yellow fever virus, West Nile virus, and there are some other types as well. So this is not a comprehensive list. This is just some buzzwords, buzz names. And these specific viruses that I'm looking at are transmitted by mosquitoes. There are also a subcategory that is transmitted by ticks, but I will not be talking about them. And basically, because they're transmitted by mosquitoes, the viruses need to be quite adaptive to survive in human cells, but also in the insect cells. This is how they look like. They're fairly small, 15 nanometers across. They're RNA viruses. And as you can see, I hope you can see my mouse, but as you can see, there is a whole kind of densely packed envelope on the surface, which is also well organized, which you can see here, the icosahedral organization of the envelope. What you can see on this representation is that it actually contains the nucleocapsid that is in the middle, that is unstructured. But most of the focus that I'll be talking about is the envelope part, which is basically this tightly packed layer of proteins on top, and then we have a membrane underneath. And that the main function of this envelope is basically to protect the nucleocapsid, to initially recognize the receptor on the cell it's going to invade, and to also conduct endosomal escape. We'll come back to that one. So basically, these viruses are maybe not as relevant in Europe, but they're definitely becoming so. So far, the infections are mostly localized around tropical and subtropical regions, but these are also one of the most populated regions in the world. So we are talking about 390 million dengue cases each year. So we're talking here just about dengue, not even about other fly viruses, but because dengue is kind of the most prominent member of the family. Around 100 million are symptomatic, and a billion people actually in risk of being infected. So we're talking about a massive global problem. As the climate change progresses, the range of mosquito is also increasing. So we are now also seeing some dengue cases in Italy and in France. So if you think it's irrelevant, it's going to become relevant very soon for everybody. So this is our boogeyman, the fly virus or dengue virus, depending on which one you want to focus on. So the main motivation, kind of like the global goal, which I will absolutely not talk about at this point, is to prevent fly viral infection. And because we're talking about viruses, I can assume that first thought that everybody has, well, why don't we just go down the vaccine route? Because that's been well established, right? There is a problem with the vaccines, and specifically vaccines against fly viruses. So this is your usual course of infection. It doesn't, we don't need to go through all the details, but basically the main point is you have your primary infection, your body starts, it basically develops immunity against whatever the antigen is, whatever your infectious particle is, in this case, a fly virus. It will develop the antibodies, and after the infection, it will create memory cells. So the next time you have the secondary infection, you would have this very quick production of antibodies. So the second infection can be shorter and can be milder. And this is exactly how the vaccines work. So the vaccines are basically going to simulate this primary infection zone in a safe manner. So you develop the antibodies and you have it in your memory cells. So, the second infection is going to be much, much milder. However, with the fly viruses and with dengue virus specifically, when it comes to the secondary infection, instead of the infection getting better or being milder in symptoms, you get something like hemorrhagic fever, or your chances of dying are much higher than during the first infection. So this paradox is something that needs to be examined. It seems that this is linked to something called antibody-dependent enhancement of disease. Specifically, it seems like the antibodies by themselves are causing these worse symptoms when it comes to dengue virus. And specifically, this has been linked to antibodies that are maybe imperfect in binding to the envelope. For dengue, that is very common because we have 4 serotypes at least. So 4 serotypes that are co-distributed in space and time. So they basically circulate in the same population. So it's very expected that if you get infected with one strain the first time, it can be expected that you might get infected with the other strain the other time, in which case you have this risk of developing hemorrhagic fever. So obviously, then vaccines may not be the best solution if the vaccines are going to develop those antibodies that are the problem in the first place. So the second solution is maybe to look into antiviral drugs. However, there's also a problem with antiviral drugs because with them you can target a lot of stages of the viral life cycle. The one we've been focusing on, and the research specifically in Singapore that I've been doing has been focusing on, is the entry stage of the viral life cycle. So over here. So when the virus is in the extracellular space, and as it enters the cell, it basically gets encapsulated in the endosome, and then it finds itself here. And as the endosome is acidifying, you can see that it undergoes some massive conformational change, and it goes from the smooth conformation to the spiky conformation. And this is what is going to help the virus escape the endosome. So if you think that's a bit of a foreshadowing, you're very correct. It is. So if we are kind of focusing on the first stage of the viral life cycle, basically what we're focusing on is the envelope, because the envelope is the only thing that is accessible at that point. Now, the problem with the envelope is that the conservation, especially the conservation of the surface exposed residues, is very low. And this is absolutely by design. So if you look at the underside of the same protein, so the side that is facing the membrane is not exposed to the immune system, you can see the residues are fairly conserved. But as they get exposed to the surface, they mutate rapidly. And this is an evolutionary mechanism of the virus, which is called immune evasion. But basically, every residue that is not functional is just going to mutate, and show different pattern on the surface. So the antibodies cannot perfectly bind to the virus. The virus is constantly escaping body's attempts to develop complementary antibodies. And this is a well-documented case, and basically the evolutionary pressure is going to drive this mutation. So you are not really dealing with something that is uniform on the surface. It's something that's more like this. It's more iridescent. It changes all the time. I've generated this using AI, but this is too high production, so we're just gonna go with this one. So basically, this is our virus with the changing surface. Right. So we have a virus that has a changing surface, and we want to use antivirals in the entry stage, for example. If we just target the surface, that is not really going to work. For once, the surface is variable between strains, as we've established. So if you want an antiviral that is going to bind to one strain, it's probably have to be different than antivirals that bind to other strains. So suddenly you need these strain-specific drugs. And if you have 4 dengue covariants that circulate in population, you suddenly have this massive number of drugs that you have to design. But even that is not really going to solve your issue, even if you manage to make strain-specific drugs, because those residues on the surface are also not going to be linked to function, especially the ones that are going to mutate all the time. So very quickly, you will develop drug resistance. So that's maybe not the best way to go about this. Instead, we are focusing on the pockets. And now this is, I mean, it's a very simple idea, basically. You can imagine the pocket, that the surface of the pocket might still be variable, as shown here. But then once you look into the pocket, because the inside of the pocket is not exposed to this evolutionary pressure, it is going to mutate slower, and it's going to be more consistent among strains. So immediately, we are working with more conserved residues. So definitely something we want when it comes to drug design. However, we still have this issue of, if we have a drug that's going to inhibit the virus, if it's in any way, if the residues are in any way non-relevant in the exact type that they are, they might also mutate further, and then you would get resistance developed. So in non-functional pockets, this will be a problem. However, if you then have the variant where you have functional pockets, that might be a benefit to you for designing antivirals, because suddenly the virus can't really mutate specific residues, because by mutating those residues, it will also destroy the function. And just to reiterate, the function of the envelope is to recognize the receptor and also to conduct this escape from the endosome, which is clearly pH-dependent. Right. So this is the methodology I've been doing. So you take your virus, you flood it with benzene, which in practice looks like this. I'm not using the whole viral particle because I don't have infinite computational resources, so I'm using a portion of it. And again, flooding it with benzene. Benzene is going to bind on the surface. And then hopefully some benzene is also going to end up in pockets. And those benzenes are going to stick in the pocket, then you can see where they are, and they remain open for longer because they're filled with benzene. So in practice, something like this. These are some practical details. I've done the simulations on 6 different viral types, 4 strains of dengue, yellow fever, and Zika, because they all somehow relate to each other. And I've used quite high concentrations of benzene in this case. The reason why I've used benzene is, it's a long story, and we can talk about it for longer, but it's kind of relevant for this. Basically, it's because it's a common drug moiety. So it maybe makes sense to flood the system with something that would also be a part of a drug. So see what the complementary surfaces are. Secondly, it is hydrophobic. And from experience, we know that the cryptic pockets that we are specifically looking for are also mostly hydrophobic in nature. So it makes sense, right, to use also a hydrophobic fragment to flood the simulation. There is a bit of an issue when it comes to having also a membrane in the system, because there's nothing that benzene likes more than going straight into the membrane and just staying there. So there had to be some special treatment, but that's maybe for some other time. All right, so just to kind of see if this methodology works, or if this approach works, if you add benzene to your system, you would expect that some benzene is packed into the pockets, that they kind of nest into the slightly hydrophobic areas of the protein. You expect your solvent accessible surface area to go up. So far so good. That's exactly what's happening. So if you have a control system without benzene, the solvent accessible surface area is lower than something that you will observe in the systems with benzene. However, there is a danger in this whole thing, where if the system contains too much benzene, then you also might get some denaturation, because benzene is a denaturant. So this was also part of the verification control scheme, to count how many structured residues that we have. So basically residues that are still participating in a secondary structure. And this may be pretty happy, because there is a very slight decrease from your experimental values and from the control simulations. But considering that we're talking about less than 1% decrease, I was happy that it was working and not denaturing the actual protein. All right, so we're going to the pocket part of this talk. I will just kind of orient you in space. This is your raft, the thing that we use. And then if you look at it from above, you can see that it has these 6 chains. Actually, there are 12, but let's say 6 for now. And so if you take the central 2, you can kind of see them over here, right? So I hope that kind of makes sense to what I'm showing. These loops that are circled are called fusion loops, and they are very important for the virus to actually do the escape from the endosome. So basically what they do, the virus undergoes conformational change, the fusion loops get exposed, and then they insert into the endosomal membrane, and they fuse the endosomal membrane with the viral membrane, and then the whole nucleocapsid can enter the cytoplasm. So they're the functional elements of the envelope protein. So if you look around the areas, again, this is the fusion loop. These are the 3 domains in 3 different colors. And you'll see some very interesting density here, at least it was interesting to us. So if you look at it, there is a pocket that has been formed in benzene simulations. This is not present in CAR-EM structures. It's much smaller in the control and very transient. And the colors here, which I also think is quite interesting, is this is the conservation of residues. So you can see how exactly the point I was making before, that the surface exposed residues might be unconserved. So red would be unconserved residue. And then as you go deeper into the protein, as the evolutionary pressure for mutation decreases, your residues also increase in conservation. So when looking at the pocket, there's also something interesting here, which ended up being histidine 144. And now I'll do a bit of a throwback, because 5 years ago, there was Proteinology Society conference in Belgrade. And at that time, I actually presented a very short talk with a deceptively similar title as of today. I promise also did some other work, not just that. But yeah, at that point, I was a first year PhD student. And I had some very preliminary results. Basically, I was looking at some the differences in pKa's for 2 static structures, the pre-fusion and post-fusion, and then looking what might be relevant, what might be not. And at that point, kind of the main outtake of the 2018 was this cluster of residues. because we kind of saw that there is some interesting clusters with interesting pKa values, and it seems like all of them are interacting with each other. And as you can see, there is this histidine 144, which again popped up now. So immediately it piqued my interest from this completely different angle. So yeah, this was something that I definitely want to explore a bit more. At the time of the conference, I also kind of presented this slide. I'm sure everybody can remember this from 5 years ago. Basically, there are some mutagenesis data about what residues in flaviviruses might be relevant for this pH dependence and for this endosomal escape. And because the way there is this, I would say, even dogma that previously been much more followed than as the time goes on. I think everybody has moved on from this model. But there's been this dogma that if you're looking at pH dependence and pH sensing residues, you might be looking at histidines. So basically what the authors did in this experimental paper, they just took every conserved histidine and mutated it to everything possible. And so they tried to see what's happening with the viral fusion. So you see that with some double mutants, you have prevention of fusion. With the single mutants, you have prevention of fusion. And this histidine 144 was annoyingly not part of the data set because they could not get a stable E protein expression. So we couldn't really say, because of the lack of data, if this is something that is relevant or not. And since then, I've also done some more lit research. And apparently there's another paper from just a year after, very interesting, which basically shown that you can get the expression of, in this case, it was also a West Nile virus. So you can get the system to express, and you can measure how infectious your sample is. So you have your wild type, or you have your histidines mutated, and with 2 different temperatures. And what you can see here clearly is, if you mutate the histidine, yes, you get very much a drop in the infectious unit. However, it's not abolished. So you still get an infectious part. So the virus can still undergo this pH-dependent conformational change. It can still escape the endosome, escape degradation from the lysosome, just less efficient. So this is kind of stolen from the abstract, but I think it's a very good point. Basically, they're just saying that this single histidine being used as a pH sensing residue doesn't seem to make much sense in the context of data. And they are suggesting that there are some additional features of the envelope that actually the conformational change. Right, so back to the present. So the question was, okay, we kind of saw from these static pKa calculations, predictions that the cluster might be relevant. But the question is it really that special? So I kind of did a very agnostic analysis of what could be a potential pH interesting region of the protein. So when we're talking about residues that might be pH sensing, obviously we're talking about residues that have to be titratable, right? Otherwise, the whole proton binding doesn't work. The second aspect that we're looking at is that they probably need to be conserved because the residues are very much functional, and we are looking at the functionality related to the residue side chain. So we expect it to be conserved among, in this case, flaviviruses. And the last one is we would like in these pH sensing residues them to be a bit buried. Basically, if you have a residue that is interacting with the solvent, it's probably not going to dictate your conformational change. You want something that is interacting within the protein, preferably something that is interacting on the domain interface. But in this case, we use a very rough estimate of just solvent accessible surface area. And then if you look at your high scores, in this case, the low scores, so the most buried residues and the most conserved ones, You see some non-titratable points, we ignore those. And then you see some on here that might be interesting to look at further. And then if you actually put this on the structure, you end up with this very, very interesting distribution of interesting residues, which again, you get this cluster. And the cluster is that all of it seems to be interacting with each other, and then these two, which are on the side protein. So yeah, this seemed to be interesting, right? This is also just showing how the conservation looks like among the ones I've actually looked at in simulations. So you can see all of them are fully conserved. So what about pKa is of course the next question, which we can discuss if it's relevant or not when it comes to buried residues. So because I was doing this research while I was a PhD student with Jim Borica, so naturally ended up using his pKa prediction method called FDDH. So it was just a method to roughly estimate what is the pKa of all the titratable residues in the system. So these, the highlighted ones, are the residues from the cluster, and there are also some others that's been mentioned in the mutagenesis analysis, which are also included here. And as you can see, some are all over the place, some are kind of fairly consistently what you would expect from the residue of that type. But the main issue with this approach is that your pKa prediction is only as good as your static structure, which in my case, I'm working with 3.7 angstrom cryo-EM structure of a virus, so maybe not the best. And another point is, because you have these buried residues, then also these pKa prediction methods also become very dependent on the local environment and the conformation of the protein. So using the static method only seemed to be a bit of a wrong approach. So, as I mean, a lot of people here would probably do the same, we kind of went in the direction of constant pH simulations, because this allows for this assessment of protonation states in dynamic structures, basically relating the protonation and the conformation at the same time. And in this case, I've used Swill's method, which is kind of a combination of explicit solvent, implicit solvent, where in explicit solvent, you're considering your conformational exploration. And then the simulation is stopped. It's implicitly solvated. The protonation states are checked and attempted protonation exchanges. And then you continue simulation and so on and so on. So this is done over a long period of time. And for this one, we've used only the active domains, so a smaller system. And these are the practical details, basically in the range of pHs with residues within the cluster that are allowed to titrate. This is just to show the convergence of the pKas over time. All right, so in terms of what we actually got, so we are talking about this cluster of charges. So it contains your arginine, your N-terminus, both of which should be positively charged. And because the endosomal conformational changes relate to low pH, we don't actually expect a flip of them, of either of them to the neutral charge. Of course, not for arginine either. And then you have histidine, your aspartate, and your glutamate. So these three kind of the main players, the relevant ones. This is the starting charge, where these two are negative charge, this is 0, so in total you have 0. And the cluster seems to be fairly stable. They seem to be interacting with each other and forming this tight unit between each other. So if you then look at all the constant pH simulations, you can relate your pH value versus the charge of the cluster. Now it's a bit of it's quite a lot of information here, but what you need to know is basically to reach cluster charge 0, there is one pattern of protonation, which is this, that is relevant. For cluster charges 1 and 2, there are multiple ways you can reach that cluster charge. So these show kind of different patterns, all of which are going to, in total, make your net charge 1. Then you have net charge 2, which are these 3 combinations. And then finally, your net charge 3 is also reached in only one way, in which case this is plus, and these 2 are 0. So this is not surprising. As you lower your pH, you also change the charge of your cluster. So the question is, what actually happens with the structure of the cluster? And this is where it slightly gets interesting, maybe also expected, but interesting. So here I'm measuring how disrupted the cluster is, based again on different charges of the cluster. And I'm also showing this granular model where basically each pattern of charge is presented in different color. And I hope you can see here clearly that the radius of gyration goes up as you increase the charge of the cluster. This is not shocking, right? If you have tightly packed +3 charge, you would expect it to come apart. What I find interesting is that this coming apart motion becomes relevant even in this +1 charge range, where you can see that the distribution it kind of shifted towards more removed cluster, and then of course as you increase the charge, it's more and more removed, so the radius of gyration increases. So the cluster is more disturbed as the charge accumulates. But even here at +1, you can see that this disturbance is starting to happen. So in practical terms, I kind of want to see what does that actually mean in terms of cluster behavior. So I took every distance between every residue of the cluster, did some PCA analysis, and here you can see what's actually describing those PCAs. Not that relevant. Basically, what is relevant is that you get a distribution of different cluster conformations, and most of the clusters are going to be in this tightly packed conformation, which is this charge 0, not unexpected. So this is your starting structure, let's say. It looks like that. And then as your pH drops down, you start to get these clusters that start dissociating from each other because the charge is accumulating. And then you basically get, so this is 0. You basically get this line over here, which is interesting, where you get your arginine that's completely pointing out in case of +2, or in this case, in case of +3, where you also get this disruption of the cluster. I'm not saying that this is a definitive behavior of the cluster. I think what we're seeing here is just on a relatively small sample that the cluster does dissociate, but it probably can dissociate in a lot of different ways. And also, if you include the whole viral particle, I assume that the dissociation is going to be a bit more controlled. But the general outtake is that you do get the cluster that is dissociated. And this is also kind of showing you that as you accumulate the charge, again, your conformation, so these are the disrupted conformation, this is the starting conformation, they kind of started going up. And you start seeing this increase as your charge goes up. So all of this to say, we get a cluster that is disrupted. Maybe not that unusual, but also what is interesting to note is that the pocket and the cluster is between the 3 domains. So if you have this disruption of cluster, what you also get is domain disruption. So yeah, So we're now looking at this domain 3, which moves out as your charge increases. And we know that for the pH-dependent conformational change, this movement, this dissociation of domain 3, is something that is a part of the process, basically based on the starting structure and the post-fusion structure. We know that the domain 3 needs to move away. So, promising. And in a similar manner, in a different example with a different pattern of cluster charges, you also see this domain 3 that is distorting itself and removing as your cluster integrating. So yeah, and right. So basically, to conclude this entire story, we have this cluster of charges that is found on the domain interface on the viral envelope. It seems to be conserved. And I think what is the most interesting outtake from this is that we're not talking anymore about only histidines that are pH sensing residues, but it's a histidine that acts in concert with other residues as well. Because we know experimentally that histidine is not be-all, end-all. So even if you remove this histidine, you still can get kinetically not as efficient, but you can still get this post-fusion conformation. And this is due to basically this redundance in the cluster, basically, which means that we can reach in ideal conditions the +3 charge, but that is not necessary to start the conformational change. So you have your aspartate and you have your glutamate to kind of compensate, and so they can get protonated and increase the charge accordingly. So if you have this redundant charge accumulation, it basically means that the virus has more fitness, even in the face of mutation of some essential residues. When the charge accumulates, I think I've shown that we do get cluster dissociation. And because the cluster is located on the interface of domains, we also get domain dissociation. And in general, this is a more kind of comprehensive fail-safe mechanism of the virus that is going to dictate in your pH-dependent conformational change. In terms of what are the implications for the drug design, which is kind of the initial motivation, I guess, of the whole project, which stopped being the drug design project, became a pH-dependent examination project. So you have this pocket over here that is touching this cluster of charges, and the pocket is conserved. I know also from comparing with between 6 different viral types that it's similar. So it's conserved in sequence, but it's also similar in dynamic behavior. So hopefully we have something that is fairly conserved within the Flaviviral family and within strains. And as mentioned before, it's most likely that this cluster is functional, as shown. So if you have the functional cluster at the bottom of the pocket, they are less likely to be mutated, at least not all of them, which means that the escape from the drug or drug resistance is a bit less likely. So just thinking about what could be a potential approach to this is either to have something that will prevent pocket opening in the first place and kind of gluing the entire structure together and the domain interface together. So the accumulation charge is not going to lead to this domain dissociation, or a drug that would affect the actual charge accumulation by itself. So yeah, that's just kind of thinking about the direction of what are the actual practical implications of the project. So This has been done in collaboration between Manchester University, so with Jim Warwicker, who's been the main driving force behind the whole pH dependence and looking at what are the implications of the cluster of charge, and the Bioinformatics Institute in Singapore, where these people have also my PhD supervisor. We're working together on the project, and as you can see here, they really like the bridges. And then since then, I've moved to Aarhus University. So if anyone wants my contact, here it is. And in this case, we don't have bridges; we have tunnels now. So you should be right. Yeah. If here again, that's the actual article. If you want to read more, and yeah, please ask if you have some questions.
Thank you. So I will start with a question actually right away concerning the computational identification of cryptic pockets. So we are doing a different but similar in aim thing that I will be talking a little bit about in my presentation. But the question is, when you use the mixed solvent with benzene, how many putative cryptic pockets do you find, or you just found the one that you selected? So do you have many candidates, and then you have to do some further analysis like conservation of residues in order to figure out which one could be the most promising, or how many you get?
Yeah, this is a massive problem with flooding simulations that I've found. Is there are so many, I wouldn't say false positives, but there are so many binding sites for benzene in my case, that you basically get the whole surface, of your protein covered by benzene, and you get breakage, and you have your side chains that are moved away, and you get a lot of these mini pockets that are forming. So yeah, I can fully see why this is a problem. And for me, I kind of ended up doing density analysis over quite a lot of, so not just density analysis within the strain. So I would basically calculate pocket surfaces with, I used MD Pocket, but whatever pocket mapping software you have. So I would calculate the density of those, and then I would do intersect with all of the data for the next strain, intersect all of the data with next strain, on next strain. And at the end, you get, you basically filter out all the pockets that are a bit transient, and you get something that is a bit more consistent between all of the strains. So that helped quite a bit. But looking at a single strain or looking at a single repeat would absolutely not bring me to, actually identifying anything.
Thank you. Any more questions?
Thank you. Hi, very nice talk. I wanted to ask you, how did you calculate the pKa convergence over time? Because when you calculate it based on probability, you need to have a lot of time dependent, and you divide one by the other, and you see, oh, this time it was 50% protonated and 50 deprotonated, so the pKa is this. How did you do that over time?
Oh, I used Windows. So basically for the constant pH I had 4 repeats, and I had every pH was ran for 20 nanoseconds. So I would calculate the predicted pKa in the interval of 1 nanosecond. Then I would predict into interval of 5 nanoseconds, then 10 nanosecond, 20 nanosecond. So kind of have the average, the predicted pKa based on the data from starting from a short time span to longer time spans. Does that make
okay? So no over like exactly equal chunks of time. You did one, then another that's bigger, and the other that's bigger, the other that's bigger, until to get to the end of the simulation, like this?
Yes, yeah.
Okay, thank you.
Okay, next question. Marilyn?
I don't know if this is a question, but we've been working on clusters of residues and seeing multiple protonation tautomer states. So I was really excited to see that you were doing a similar analysis. And we're doing this in very different contexts in proton transfer proteins, where you don't really have a binding site. You just need to get the proton, from point A to point B through some intervening thing, and finding that the clusters really reduce the conservation. So if you have a single site, it's really conserved. But if you have a cluster of sites and you look at them genetically, sometimes they're poorly conserved. So since you did the genetic analysis, could you talk a little bit about the cluster conservation? Because I think what you're saying is the cluster makes it robust. There are lots of ways to get the same result. And that I think should show up in sort of, a softer conservation index.
That is a very good point. So in this case, what I'm seeing is that the cluster is very conserved. So while I was doing the cluster analysis, it seems that it does seem to be very robust. So I think I showed you the alignment, right, for the dengue strains, yellow fever, Zika, but basically the cluster remains as such. And then of course you have the N-terminus, which is not conserved, but I don't care, right? Because the N-terminus is still pointing the same way. So yeah, I'm not seeing per se that the cluster is more ready to mutate in population, but I am, what we are seeing from the experiment, that the virus remains viable in kind of the experimental condition. So the mutation of histidine, for example, still keeps it somewhat functional. So I'm wondering maybe if this is also the effect of the fact that the cluster is buried, so there is no really pressure, evolutionary pressure for the virus to evolve away from the system that is obviously working, because it doesn't seem to be a target for the antibodies. And because it's not a target for the antibodies, it can just remain as is. And we do see that, the moment you mutate away, even within the cluster, you do get this reduction of infectivity. So I guess it would just filter out from the population fairly quickly if you do get the mutation in the cluster. But that's just speculation.
And another thing that a cluster does is you don't get the same kind of pH isotherm. Like it can be sharper, or it can be broader. So again, you did the pH. How does the pH dependence of the total charge? Is it steeper or shallower than, what's the N value?
I don't know. That I genuinely don't know the answer to the question, but that is a very good point to look into.
Okay, there is one more question.
Hi, I wanted to ask you, especially after you gave the answer about filtering out the transient pockets, I've seen cases where transient pockets were the pockets that you were actually looking for, and they were very rare. And so how do you manage, or how do you know a priori that you should be filtering out these transient pockets? Because maybe they could also give you a hint into important cryptic pockets that you're not really looking into by filtering them out.
So the main filtering is happening. So I'm keeping. Okay, so the main filtering is happening between the strains. This is kind of the advantage of running 6 different strains, that you expect to have at least overlap in these transient pockets. But then when it comes to the individual densities for pocket mapping, I'm keeping it rather loose. So when I'm looking, for example, for single strain, I'm putting the frequency of the pocket relatively low. So I have a lot of pocket on the surfaces. And then as I'm filtering it out among the strains, basically I get the pockets that are, even the ones that are appearing in shorter time spans, but the ones that are appearing between the strains are kind of the ones that pop out, which is exactly the ones I want to look at. Does that make sense?
Okay, thank you.
Okay, mine is more of a comment, I think, than a question. But you have such a nice example here that I think it's a good example to make this point, which is when you have a cluster, like in your case, and when you have these various possibilities, these various configurations and combinations of protonation states that can happen at a given pH, for example, they have, this corresponds from a thermodynamic point of view, this corresponds to a high entropic contribution to the process. And the way I'm saying this is because people sometimes forget this and they insist that entropy is only related with motion. And in these cases is not. And cases like yours are an excellent illustration of that, because you would have the several possibilities. Is like the motion of a side chain, for example. You have the same charge, but you have the different combinations, and that gives an entropic contribution to the process, which I think it's always very interesting. And your case is an excellent example of that, I would say.
Thank you very much. That's a comment. I did not think about in those terms, even though, yeah, I agree the data does kind of correspond to that.